There's our cow. There we go. It's a Holstein. I don't know. The cow's name was Blossom. So Blossom gave cowpox to Sarah Nelms. They took Sarah Nelms' cowpox, gave it to James Phipps. He got cowpox. They challenged him and deliberately exposed him to smallpox, and he does not get smallpox. This is called variolation. It is not quite the same thing as vaccination. It's a very crude form of vaccination, isn't it? But let's think about the word vaccine and vaccination and where it comes from. And if anyone here speaks Spanish or Italian or Latin, what's the Spanish word for cow? It is vaca. Well, that's Latin for cow. Vaca is cow. And therefore we call this process, vaccine, comes from from the root word for cow. So, if you're ever on Jeopardy, moment of silence for Alex Trebek. If you're ever on Jeopardy and that comes up, you'll know where the term vaccine comes from. Alrighty, so fast forward to let's fast forward to the 1900s. 1900s. We all remember them, I think. No, maybe we don't. Some of you were probably born after 2000. But let's talk about the early 1900s. There would often be epidemics, not pandemics, but epidemics of a disease caused by a disease called polio. You've probably all heard it. You've probably all been vaccinated for it. Polio. Polio was a nerve infection caused by poliovirus. By the poliovirus, of which there are several strains. So it's just like COVID is caused by the coronavirus. Polio is caused by the poliomyelitis virus. And it was really a scourge. I mean, I think we all have a really good idea now of what it's like to be afraid of a virus and having it change our habits, right? It has changed all of our habits, whether you agree to them or not. We are required to wear masks indoors at stores, for example, or they're not going to let you in. We've all had to change our habits. The reason I'm talking to you here is because we had to change our habits at colleges and schools. We have to teach remotely right now. And it's not forever, but polio outbreaks caused a change in behavior in society as well. And probably none of us here are old enough to remember any of that. I'm probably the oldest of anyone listening to this now. I don't remember because the last outbreak was 1954. Polio was a mild infection for like 90% of the people who caught it, but for the 10% who weren't so lucky to have a mild infection, they developed paralysis. So paralysis of the leg because it gets into the nerve cells, and sometimes paralysis to the point where people had to be in an iron lung, which is this giant machine that pushes your chest cavity in and out to help you breathe because your lungs don't work properly anymore. Famously, an American president, FDR, Franklin Delano Roosevelt, developed a polio infection on Campobello Island in the 30s, I guess, during an outbreak there. It was contracted. It was not airborne, it was waterborne, and he was swimming in water that somehow was infected with it, and he was quite famously disabled and quite famously hid his disability from the American public because that was not an okay thing to be out in public about in the 40s. Anywho, where was I going with this? But back in the 50s, my mother would say, oh, no, I remember swimming pools being closed and lakes and beaches being closed because you could not swim. it traveled through the mouth and fecal route as well. So contaminated water, we have really good water treatment plants now, but if water were contaminated at all, not properly treated, it would get into water. It could get into a large water reservoir, for example. So it was a big, hairy deal. And it was I'm trying to remember my dates here. But there was a huge push to develop a modern vaccine because we had this variolation thing here for smallpox, and we didn't have anything that worked for polio at all. But a researcher named Jonas Salk at the University of Pittsburgh, Pittsburgh Medical School, developed a vaccine, and I want to say it was actually released in, oh my gosh, let me see if I can find it. I should have had the internet open for this. I want to say it was like 1957, not sure, but so late 50s, they released the first polio vaccine. And it was a game changer. So you know how excited we are now about the potential for not only one, but 2 vaccines against coronavirus. There was so much rejoicing. I mean, Jonas Salk won the Nobel Prize for this. He also went on to form the Salk Institute for vaccine research. It's in La Jolla, California. Right. But he also started a research institute at the University of Pittsburgh Medical School as well. But it was a game changer, and the first polio vaccine changed public health as we knew it, because vaccines are one of the biggest tools that we have in our public health toolbox. So we're going to be talking about the different types of vaccines that we have available to us. So first up, what is a vaccine? Okay, we talked about smallpox vaccination. It's really variolation. Vaccine, definition: preparation of a pathogen or its products that is used to induce active immunity. I want to add one little thing to this, so I'll take a screenshot here. A preparation of a pathogen or its parts that is used to induce active immunity. Active immunity where it gets your body to make its own antibodies against it. All right. So first up, let's talk about live attenuated vaccines. Sometimes this is called whole agent, or I take that back. These are called live vaccines a lot of the time. This is where we give someone the virus, but it has been attenuated. This term attenuated means weakened, not weekend, as in Saturday and Sunday, but weakened. A live vaccine that is attenuated, it means the virus has been weakened to the point where it can still get inside your cells, but it can't cause disease. So here we go. I'm just writing this again, even though it's already written here. Patient is infected with a weakened form of the pathogen such that it does not result in disease. We're crossing this word generally out just because everything has a risk. And it's called a live vaccine because it's still the whole virus. I don't want you to think this is coronavirus because this is not what a coronavirus vaccine looks like. I'll just make this one look a little different. I'll put an icosahedral in here. There we go. We'll take, let's say this is infectious. This is the real thing. What we will do is either genetically engineer it or grow it in the laboratory under certain conditions so that it gets weakened. Or, ooh, better. I'm going to erase all this. Ooh, better. Oops. Like, let's say what it does, like it's still alive, it's still a virus. Viruses aren't alive, I know you know that. But maybe it does something to the little spike proteins that are on it to the point where it can't make you sick anymore. So it's attenuated. And here actually, or like messes up the protein code or something in there where it can't cause disease. Attenuated. Can't cause disease. Something like getting cowpox instead of smallpox, only even way less than that. Way less than that. Where maybe it just gives you the sniffles for a couple days or something like that. An example of a live attenuated vaccine is one that I have had. I'm going to delete that. And that is actually the oral polio vaccine, or OPV. This is not Jonas Salk's vaccine. This was made by a dude whose last name is Sabin. I don't remember their first name. But the oral polio vaccine came out a few years after the first polio vaccine, which we'll talk about in just a minute too. Oral polio vaccine, it's a live vaccine. Live vaccine. Oral, because they basically put a drop of it on to your mouth, and we gave that to children until 2000, the year 2000 here. So there might be many of you who have not had the oral polio vaccine. But that's one example of it, of a live attenuated vaccine. All right, so attenuated vaccines, live vaccines, got some major advantages. There is a reason why live vaccines are basically the gold standard for vaccines, and that is because, okay, it does not reduce in disease, but one dose can induce immunity for a long time, for a lifetime sometimes. That is huge. That's huge. Getting a vaccine and having it last a lifetime. You don't need booster shots. If you've ever had a tetanus shot, you probably have. You got to get one of those every 10 years. Live vaccines, live attenuated, not so. They are the gold standard because they actually infect a person. They cause an infection, which doesn't make you sick. And by cause an infection, I mean they get inside your cells. You get a really strong immune response. The measles vaccine is an example of that. Is a live attenuated. And you get such a good response with that, which is a darn good thing because measles is wildly contagious. Let me think. Measles, the shingles, and the chickenpox, and the oral polio. Those are the only ones I can really think of right now. But the measles one, that is huge, and it is hugely effective. You don't need to get boosters for that at all. Patient is infected with a weakened form, so it doesn't result in disease. One dose can induce immunity for a very long time. All right. A productive infection means a stronger immune response. Already said that. Now, this last bullet here is kind of interesting. Theoretically, because you actually get an infection, the fake measles virus, the attenuated measles virus gets inside your cells. Technically, you could, pass that on to someone else. Now, we have not seen evidence of this happening. Like, you could passively give the vaccine to someone else, but we have not really seen much evidence of this. But theoretically, it could be happening, and that would be quite an advantage, too. Okay, so attenuated live vaccines pros, got those. Cons, because they are a live virus, they can mutate and become pathogenic and cause disease rarely, if ever. It's a very low risk, but it's a risk nonetheless. I do not know what the statistics are for the measles vaccine, like how many, does anyone ever get the measles from it? I know that it is very low, and I know I could go on and check on the World Health Organization's website to see what the current data is, because I will say that the CDC, the FDA, and the World Health Organization, when a vaccine has been created, once it's been approved and has been in use for a while, they don't just write it off, say, okay, that's one and done, we're done. No. They continue to monitor the effectiveness and any complications to a vaccine. They're constantly looking at the effectiveness and risks of every vaccine that's been approved. So they got our backs. It's not a one and done. They continue to look at those. All right. They can cause disease in immunocompromised patients and in fetuses. Pregnant women in general cannot receive a live vaccine because they can pass it on to a fetus. So if other patients who have certain autoimmune disorders or somehow immunocompromised based on medications that they have, or someone who has who has an HIV infection and has already shown signs of developing acquired immunodeficiency syndrome, they can't get live vaccines. And we'll talk a little bit about this later too, but when we discussed herd immunity, we want everyone, we don't just get vaccinated to protect ourselves. We vaccinate to protect an individual. I don't get a vaccine just to protect myself. I do it to protect the people around me too. People that I don't even know, people that I will run into in the grocery store who might be immunocompromised and therefore unable to receive a live vaccine, for example. So it's to protect others as well as to protect me, because some people cannot get certain vaccines. All right. This is kind of a wishy-washy downside, but because there are other vaccines that are not live vaccines that do require refrigeration, and we'll talk about that with the coronavirus vaccine later on as well. But most live vaccines all require refrigeration, which kind of might be a bit of a drag when it comes to trying to vaccinate, massive numbers of people all over the world, which is what we're going to be doing with the coronavirus vaccine, with the COVID vaccine. So this is kind of a downside, but other types of vaccines have that too. So can't really worry about that. Okay, so 2 general categories exist for vaccinations. Let's go with. We haven't done a different color for a while. Let's go with blue. In general, there are live attenuated versus all the other types of vaccines versus, let's say, inactivated. That's what your book categorizes them at, but I might actually have you watch This Week in Virology thing in which they don't categorize all vaccines as either live or dead, live or inactivated. But there are many different types of inactivated vaccines. The first of which is what we call inactive whole agent. And an example of this is the Salk polio vaccine. The Salk vaccine is an inactivated whole agent. So by whole agent, I mean the whole virus. But it's been inactivated to the point where not only does it not cause disease, it can't get inside your cells. So let's do a little graphic here, and I need other colors. Okay, whoa. Ooh, let's go with red, Dayglow red, and say, okay, here's a virus. Right there, and let's say it's got spike proteins here. Let's say this is live infectious virus, not live attenuated, but live infectious poliovirus. I don't even know what the poliovirus looks like, so I'm just drawing something here. We treat it in the laboratory in such a way, not grow it, but we treat it with chemicals, like it's often formaldehyde. Something that basically does a number on the vaccine, on the virus, so that it's intact. You still have the whole thing, but it's dead. Its spike proteins are like, something's happened to me. I'm all crappy now. Now I can't use my spike protein to bind to some receptor on this human cells. I'm dead. I am inactivated. It can't cause disease because it can't get inside your cells. BC, because it can't get inside your cells anymore. Which means it does not cause an infection. Not only does it not cause disease, it doesn't cause an infection. Whereas back here with the oral poliovirus, live attenuated, I've got to go back here. It still causes an infection. So here we go. This is what I want. Patient is infected with a pathogen such that it does not cause disease. Okay, so heck with this. I'm going to go back to my notability. Oh, God, stop! I'm pressing the wrong buttons here. Okay, inactivated can't cause disease because it can't get inside your cells anymore. It doesn't cause an infection. Live vaccines, live attenuated, cause an infection, but without the disease. So live attenuated causes an infection, but inactivated doesn't even cause an infection. Doesn't cause an infection. It's inactivated. It's like, so how come it still works if it doesn't cause an infection? Because there's still something about maybe this or something else that's on the envelope will still cause an immune response. Will still cause an immune response. But it does so without causing disease and without causing an infection. This is the Salk vaccine. This is the first modern vaccine that we had, and that we were able to develop a vaccination program to basically rid the world of polio. The world has not been completely rid of polio, however. There are some pockets of it. I believe the goal was to end polio, annihilate it by the year 2000. We did not. I think it was 2007 when it was nearly eradicated, but there are still some pockets of it now. Now let's look at the pluses and minuses, the pros and cons of an inactivated vaccine. Inactive whole agent. They don't cause an infection, so really low risk. Really, really low risk from the Salk inactive whole agent vaccine. Very low risk. You cannot get polio from the Salk vaccine. Usually they don't require refrigeration, but again, this is wishy-washy again, because some that are within this category actually will. It just depends on the preparation. Again, we'll talk about that in the next lecture. On the downside, protection doesn't last very long. So you generally need booster shots. So the Salk polio vaccine required some boosters, whereas the oral polio vaccine did not. So let's compare and contrast the pros and cons of inactive whole agent Salk vaccine to that of the attenuated Sabin oral polio vaccine. How might you decide which one to give to a community? It's, again, this is the debate we're having now about coronavirus vaccines, although I don't know if there is a live attenuated corona vaccine trial out there. I think actually there is not. But let's look at some pluses and minuses of the Salk versus the Sabin vaccine. All right, so Salk, which is inactive whole agent. And this one came out first, but just a couple years later, the Sabin oral polio vaccine came out, which is live attenuated. I misspelled attenuated. You got what I mean here. Okay, the Salk came out first, so yes, everyone in the late 50s got this right away. But, like, what do we do now? Salk is no risk, really. You cannot get polio from it. You might have some adverse reaction to something else that was in the vaccine, but it's low risk. Sabin is lower, is low risk. Now let's say slightly higher risk. Slightly higher risk. It's still low, but it's still slightly higher. That higher risk that the live vaccine could mutate and revert to its pathogenic form. Salk vaccine, though, because it's low risk, because it's an active whole agent, it's not as long lasting. Sabin vaccine could last a lifetime. I don't know the exact number of years they say, but I believe it's actually lifetime. We do not get oral polio vaccines as adults anymore, unless you're going to go to a country where it might be prevalent. So what do? Which one do you want? Especially if you're working at the World Health Organization and you're trying to figure out a way to help everybody, not just you or your child, but the entire planet, and rid it, because that is going to help me and my family. Here's what we used to do for a while, and I think I got to research this because we may have gone back to this protocol for a while. And I know we have in certain parts of the world. All right, for a long time, we would just give the Salk first. To 1-year-olds. I forget what the vaccination schedule says to give polio, but I think it's right after DPT. So you give that first so that you have some immunity to it. And while you still have that immunity, you follow up later with the Sabin vaccine. So that just in case there were any risk with the live attenuated, you're protected by antibodies that your cells already made in response to the Salk vaccine. Now, in the year 2000, I think because the prevalence of polio was so low then that we ceased to give the Sabin vaccine. That has turned out in several parts of the world to not be a great thing to only give the Salk vaccine, because here, for example, and I want to say the example that I read in your book was Israel. And I forget when, in the 90s or in the O's or even 10 years ago, everyone got only the Salk vaccine. So nobody got sick from it, but there was still a lot of live, real poliovirus around, and we want to get rid of it. We don't want it to have any human reservoirs anymore. Only Salk vaccine, so the real poliovirus stuck around, persisted. Because it wasn't challenged by people who had long-lasting immunity from an actual infection from it. So I believe they actually started giving people the Salk vaccine as usual, and then the oral. So now it's Salk, then Sabin, oral polio vaccine. These are the kinds of discussions that public health and epidemiologists have to have on a regular basis. Alrighty, so that's enough for now. Those are the basics of live attenuated vaccines versus inactivated whole agent. Next time we will talk more about the other types of vaccines, and we will talk about the 2 coronavirus vaccines that are looking promising right about now, and we will look into vaccination programs and how that has to occur. Okay, everybody, let's finish up vaccines, and this will be our classes, the full class for the week of November 30th. Anyhow, we talked last time about the difference between inactive whole agent vaccines, such as the Salk polio vaccine, and live attenuated vaccines, such as the Sabin oral poliovirus. I should add that live vaccines are really like the gold standard because they just tend to grant long-lasting immunity. You get a much stronger, long-lasting immune response. And just to rehash a little bit, or to clarify a little bit about the concept of vaccinations, we are actually in Chapter 18. Your immune system reacts to molecules that are presented to it, molecules that are on a pathogen, for example. So, let's say, and the pathogen doesn't even have to be a virus, but let's say, let's say, yes, it's a virus. Let's put an envelope around the capsid and, maybe put a couple of some spike proteins and a matrix protein that holds that envelope together. There might be some other molecules here too. This would be, if we grew this in the lab and we grew it under conditions that allowed it to be attenuated, this would be, essentially a live vaccine. Your immune system sees this and goes, what the heck is this? What is this? This thing here, that's not self. This little molecule here is not self. These lipids here, this is not self. And so through a whole series of events, including antigen-presenting cells, your immune system eventually will make antibodies, and I'm abbreviating antibodies with ABS, will make antibodies against certain molecules, certain that we consider antigens, against certain molecules, aka antigens, that are a part of the pathogen. Okay, now the presence of antibodies is essential, but that's an oversimplification of developing immunity. You actually need a T cell response as well. And so with all the coronavirus vaccines that are currently in trials right now, they are looking not only to see if patients have made antibodies and prevent disease, they're also looking at a T cell response as well, specifically killer T cells as well. But we're focusing on antibodies because honestly, that is a major source of evidence that we have immunity. So for example, there are vaccines that are not as long-lasting, and we talked a little bit about that with the inactive whole agent. It's not as long-lasting a vaccine. There are some other ones such as the hepatitis B vaccine. It is not a live vaccine. It does not last forever. And so if you are a healthcare worker and constantly potentially exposed to hepatitis, you have to, basically we say, we have to get your titers done. So you get your titers done. This means, do you still have antibodies against the hepatitis B virus? So it checks for presence of antibodies. So, and those of you who work in healthcare now, and I know a lot of you do, are well aware of this. You have to get your titers done every now and then. All right. So far, we have talked about live attenuated vaccines, and also inactivated. And just quickly, I don't mean to spin you around. I had the right slide all set up and it moved around. Okay, so we talked about live attenuated. Nope, I guess we got to go. Nope, it's forward. My bad. Here we go. There are different types of what we would categorize as an inactive vaccine. Okay, we already talked about inactive whole agent. I want to talk about these protein subunit, though. It's a subunit vaccine. And we'll go back to notability. Somebody decided, okay, so we've got this live vaccine up here, but they carry a risk or something, and really we just need something that gets a good immune response. Do we really need this whole enchilada to stimulate immunity in the immune system? And the answer is oftentimes no. That you don't need the whole agent. Somebody had the idea saying, you know what, if we can get an immune response to, say, just this little spike protein here, why should we give the rest of this vaccine to everybody else? So somebody's like, hey, y'all, what if we make a vaccine that is basically just this piece here? Just a little part of the protein. So that would be a protein subunit vaccine. And the hepatitis B vaccine that I talked about actually is a protein subunit vaccine. So protein subunit is where just a piece of one of the viral or bacterial proteins, because not all vaccines are against viruses, they're also against some bacteria too. Protein subunit vaccines are when just a piece of the protein can stimulate immunity. Piece or subunit of a pathogen's protein is enough to stimulate an immune response. That is enough. And the hepatitis B vaccine is an example of that. I want to say Gardasil is, but I'm not so sure of that right now. Not 100% sure. All right. So there is protein subunit vaccines. There is also toxoid vaccines. Okay, this term oid here means similar to or like. So a toxoid is a molecule that looks like a toxin. But doesn't make you sick the way a toxin does. So it doesn't cause illness. But instead, like let's say a toxin is, like this molecule here. I don't know, I'm just making up shapes here, okay? I don't know what's possessing me to make a shape like this. I'm just making something up. Maybe that's the toxin, but maybe the toxoid looks like this. I'm just making up molecules with shapes here. Okay, maybe this shape here is enough to stimulate an immune response, but doesn't make you sick the way the real thing, the real toxin does. All right, an example of this is the diphtheria toxoid vaccine. There is the DPT vaccine, which is D is for diphtheria. This is a combination vaccine, tetanus and pertussis. Is it pertussis or pertussis? I'm not sure if that's an I or a U. I think that's another U. I don't know. Pertussis. But the D part, the diphtheria part of this combination vaccine is a toxoid vaccine. Toxoid. Yeah. Okay. There are polysaccharide vaccines. Polysaccharide vaccines are generally not given to children. So let's just screenshot this right here. All right. Let's see. Polysaccharide vaccines are when, and this is more for vaccines against bacteria rather than viruses. So usually against bacterial pathogens. All right, there is a meningococcal vaccine. Can I spell meningococcus? Meningococcal vaccine. certain people are at risk for that. All right. So we have those. There's some, and pneumococcal vaccines that a lot of older adults will take. Pneumococcal. I can't spell pneumococcal either. All right, a conjugate vaccine. Conjugate, to conjugate means to get together, right? And a conjugate vaccine is basically a polysaccharide vaccine that's linked to a protein. Let's say attached to a protein. There have been some flu vaccines that have been conjugate vaccines. There are all different kinds of flu vaccines, though. So I don't want you to think that all flu vaccines are conjugate. No, they're actually not. A lot of them are protein subunits. Some of them are actually live vaccines. There are different ones. They change every year, and there are different choices that you have every year as well. But the conjugate vaccine, I believe the Hib is always Haemophilus influenza B. The Hib vaccine is a conjugate vaccine. Alrighty. Now the last one that I want to talk about is this messenger RNA vaccine. This is very new. If the current, it's Pfizer and Moderna, I think, are the two SARS-CoV-2 vaccines that are messenger RNA vaccines. If either one of those, makes it through and it looks like it's going to happen, they will be the very first approved messenger RNA vaccine. So let's talk about how those work, what the deal is with that. All right, so I want to say a little bit about these messenger RNA vaccines. The Pfizer and Moderna are both RNA vaccines. Now, how does this whole thing work? Okay, I know that this has been a very technical lecture for you all. Don't worry, we will clarify things and I'll give you a study guide for it all as well. But basically, and we will be getting to this part that I'm going to talk about, later in the course as well. But to summarize, you've got DNA in all of your cells. And this is actually where your genes are. Your genes are in your DNA. Okay, and if you look up the word gene in the glossary of your book, and it's actually up there at the end, there is a glossary. A gene is it's probably going to say the basic unit of inheritance. The basic unit of inheritance. But what does that mean? I mean, we know that we inherit our genes from our parents and we get our traits, from our birth parents, we get our traits somehow for our genes, and our traits are our physical characteristics, what your earlobes are going to look like, what eye color you have, that sort of thing. But what does that mean? So we need an expanded version, we need an expanded definition of a gene. So, that's just one definition. 2, let's expand our definition of a gene. A gene is actually an area of DNA that contains information. And that information tells your cells how to build a particular protein. So let's write this down. An area of DNA that contains info, telling your cells how to build a protein. How to build a protein. So basically, in other words, genes are a set of protein plans. Genes are, let's say, blueprints. For building proteins. Building proteins. They're plans for proteins. They tell your cells how to build a protein. And proteins are actually the molecules in your cells that do a lot of work. But before the cell doesn't, it looks at the DNA and it sees the genes there and it's like, ooh, there are protein plans in this piece of DNA. To get that protein made, the cell actually first makes a copy of the protein plans, and that copy is called RNA. So it's a copy of the protein building plans. It's a copy of it. Then what the cell does is it looks at the copy, because it doesn't want to mess up with the original. These are the originals in the DNA. The cell will build a protein by basically reading what the RNA has to say. Later on in the course, we will get into this more deeply, but I'm talking about this now to help you understand what these RNA vaccines are all about. So the protein is sort of the product of the gene, but the cell needs to look at a copy of this. Okay, so let me write this down again down here. So you have DNA, which is your gene. The cell makes a copy, which is called RNA. And sometimes it's got a little M in front of it. We'll talk about that later in the class. But what's most important is that this is the copy of the gene or the protein plans. And the cell looks at that copy and builds a protein from it. When it comes to vaccines, it's what your cell sees as not self, like what I was talking about before, is usually a protein. So your immune system makes antibodies generally against proteins. Makes antibodies. I'm going to abbreviate antibodies with ABS. I've done that before, but just to make sure you know, antibodies. So it makes antibodies to proteins most often. It doesn't, your immune system doesn't make antibodies to messenger RNA. Doesn't do that. Not very much. So the idea behind, for example, the polypeptide vaccine that I had already talked about, it's like we give a little protein to your cell. But these new vaccines are saying, we don't really get that great of an immune response with protein vaccines. Why don't we just give this piece just the messenger RNA from the virus? So let's say here's coronavirus. I'm going to put its little fluffy outer coating there and its little RNA in there. And let's say this is, coronavirus. Ooh, I'm going to put the little spike proteins on it. There we go. If we give this whole live coronavirus to people, that we're going to get them sick. Okay, we talked about that a little earlier with inactivated vaccines. But somebody's like, what if we don't give any of these spike protein things? What if we take that away? We take away the little outer coating. Why don't we take away the protein coat from this virus too? And why don't we just take some sort, we need a different color here. Why don't we just take the messenger RNA, or the RNA, from SARS-CoV-2. Why don't we do that? and we'll give this to people as a vaccine. Well, here's the deal. You can't just give this little naked piece of RNA as a vaccine. It won't get into your cells that way. So scientists were like, what we need to do is package up this little RNA in some sort of very benign fat molecule or lipid molecule. So let me get rid of these arrows here. And let's say we need another color here. Let's put it in brown. Okay. And we'll make some sort of little, lipid fat molecule that will package up the messenger or the RNA from coronavirus in. So this is just some sort of lipid, aka a fat coating. And this basically makes up the vaccine. It doesn't have the other parts of the virus that are gonna make you sick. It just contains its RNA inside basically a piece of lipid. It's a very, very basic vaccine. And the idea is that this gets into your cells. Let's make our cells, make them green. Here we go. Let's say this is your cell. And let's say this little vaccine, which isn't a real virus, it's just part of one, gets in there. Your cell's like, hey, this is RNA, and that's a copy of a gene. Let's make a protein from it. And so let's say it makes that viral protein, and your cells will degrade the rest of it like that. And let's say the viral protein is, I'll make it this little blue blob. Let's say that's the protein that it makes from it. Your immune system sees this and says, let's make antibodies to this. Ooh, this is not self. Let's make antibodies. ABS. So that's the gist of how these messenger RNA vaccines work. I know I'm throwing a lot of material at you. I know this is a really super long lecture, so I'm going to use part of this next lecture for next week as well. Oops. Didn't shut off notifications yet again. Yeah, so let's delve into that again. And we'll talk about what the efficacy is and how efficient this vaccine is for Pfizer and Moderna. Central dogma of biology is that, there's DNA and the gene is in the DNA and it gets transcribed into a copy, which is messenger RNA. Messenger RNA, which is the copy. And it's the messenger RNA that the ribosome reads. Ooh, I'm going to draw a little ribosome here. Like it's, the 30S and the 50S ribosome. Well, that's bacteria. I guess in humans it would be the 40S and the 60S. So there we go. There's our little ribosome I decided to draw. And it builds a protein based on what that messenger RNA says. Okay, with RNA vaccines, it is the concept here is that we will introduce the body to the messenger RNA, to this little piece of viral RNA, and that the ribosomes in your cells will build basically the spike protein from that. I'll draw it like that. If we're talking that's the little spike protein on coronavirus. Like, let's draw a little coronavirus. What the heck? Y'all are going to get so tired of me drawing these viruses, aren't you? I'm going to put its messenger RNA in there. We'll keep it coated in purple. And I'll put the little spike proteins out here. Okay, now you know that it's the spike protein that allows the coronavirus to enter the cell. We've talked about that before. this thing right here. And so people are like, there is one vaccine, but it hasn't panned out, where someone's just like, well, why don't we just give the little spike protein to people, and their immune system will form antibodies against that. And that's definitely one idea, but the general idea with messenger RNA vaccines are like, okay, the messenger RNA here inside the coronavirus, and I'm up here in the upper right, so please be looking here since you can't see where I'm pointing to unless I'm drawing something. Right. The directions for how to make that spike protein are in the messenger RNA, right? Right in there. So this little piece of messenger RNA. So Pfizer, and I believe it's Moderna, yes, Pfizer and Moderna are the ones that are working on these, are like, why don't we just package up the messenger RNA and give that to people as a vaccine? But the thing is, you can't just inject people with messenger RNA. RNA does not last very long. It will be broken down by RNases within minutes. Surely, it's just not long-lived in the cells. So they had to package it up in something, and they decided, well, we'll package it up in basically something that looks a little bit like a viral envelope. We'll package it up in lipids. And the lipid will protect it long enough for an immune response to fully develop. Now, I should add that this lipid coating is fragile. And with the Pfizer vaccine, it needs to be stored at -80. Which is kind of a problem because a lot of hospitals, places that usually give out vaccines, your regular doctor's office, CVS and Walgreens, don't have freezers that go down to -80 to store all of these vaccines that need to be stored at that low of a temperature. -80s are actually very common in research laboratories. I mean, all the labs I worked in, we had several -80s because, it's really great long-term storage. But, CVS doesn't have those, and your regular doctor's office doesn't have them. So it's a little bit of a problem. It's definitely a disadvantage, but hey, we can do this. We can do this as a world and get everybody, all these places that need -80s, a -80. The Moderna vaccine, on the other hand, their little lipid vehicle for getting it into your cells, it only needs to be stored at, I believe it's -20. So stored at -20 degrees C, which is a typical usual freezer temperature. So this one is a little bit easier. Now, both of these vaccines have come out saying that Pfizer has said it's about a 90% efficacy. And I think Moderna came out and said that they're 94% efficacy. And when Moderna came out and said they're 94, Pfizer's like, oh, wait, we looked at our numbers and we're saying we're 94 as well. So we're the same. So yeah, we've got a little bit of a competition here. But the idea with their vaccines is that the messenger RNA gets inside your cells. Here, let's just draw it this way. Here we go. That it gets inside your cells. Let me draw the lipid thingy. And, here's your nucleus and all this other stuff. And we've got ribosomes in our cells here. That once inside the cell will break down that lipid and then translate the ribosomes of your cells will translate this into the little spike protein, and then the spike protein will stimulate an immune response. And it looks like that's really happening. So let's talk for a minute about what this 94% efficacy means. I'll just use the Pfizer vaccine as an example. Basically, they tested about 40,000 people. About 20,000 got the vaccine, and the other 20,000 got a placebo, a shot that just had some saline in it. So it wouldn't stimulate the immune system. And the people who got the vaccine and the people who got the placebo, nobody knew who was who. And this is after the vaccine has been tested for safety. And what they found, and basically they had to wait around for people to get sick to see who got sick. And they basically, I think they got only something like 100 cases? No, it must be something like 1,000 cases. But 90% of the people who developed COVID had the placebo. Does that make sense to you? 90% of all of the people out of the whole 40K, out of both groups, the group that got the vaccine and the group that got the placebo, 90% of the people who developed COVID symptoms had gotten the placebo, and only 10% had gotten the vaccine. So that's what this 90% efficacy means. And that is really, really excellent news. Now, there are a few questions about this that I and lots of other people have. Okay, for testing for COVID, 90% of the people who got COVID had the placebo. Not all of these 40,000 people were tested for the virus, for presence of the virus afterwards. So the people who got COVID, this doesn't take into account anybody who was asymptomatic. So it doesn't account for asymptomatic cases. And we know there are a lot of asymptomatic cases. And what we don't know is maybe the people who got the vaccine, were they all asymptomatic? Were they still passing it on to other people? We don't know. And the other thing is, and this is less of a concern because the people who were selected for this vaccine trial were selected because they were probably at somewhat risk. They weren't people just totally staying home. Part of but still, is it like, well, if not a lot of people got sick out of this whole 40,000, is it because they were all staying home, working from home, and never went anywhere anyway? That's definitely a possibility, but I would say that from what I have read about how Moderna and Pfizer and all of these companies, all these pharmaceutical companies working on a COVID vaccine is that they are looking for people who do not work at home, who have to be out and about to work. So they were trying to select for people who, regardless of whether or not they got a vaccine or a placebo or even participated in the trial, we're going to be out in public. We're going to be working around other people, so that they could say, that it's probable that they have been exposed and they still didn't get sick. So this is good news. This is good news. The only major concern that I have and that I've read about other epidemiologists have is that this still doesn't account for any asymptomatic cases. So we'll see. But it's very, very good news. And Moderna had a similar thing going on here too. So yeah, they just had like a 94% efficacy. One more thing, this term efficacy, this refers to how well it worked in the vaccine trial. It's not the same as effectiveness. Efficacy does not equal effectiveness. Excuse me. If a vaccine has 90% effectiveness, this refers to how well it works out in the real world. Oops. Got one of my notifications coming in. Okay, so that's the reason that they're using the term efficacy as opposed to effectiveness, because efficacy is how well something works in the trial. When it gets out into the real world, we'll see. We'll see. Because then, you can't hide asymptomatic cases. Let's see. So we've got that. Yeah, and then finally, what I really need to say about this whole, about both vaccines or any of them, is that I read a quote, oh gosh, it was from some infectious disease expert who said vaccines don't save lives, vaccine programs do. Vaccine distribution programs are what saves lives. And their point was like, well, okay, vaccines actually do save lives, but they don't save lives unless you get the vaccine, right? So it's really vaccination distribution programs. Save lives. So that's why it's a major deal trying to work out how to get this vaccine out to everyone, how to get it to the riskiest populations first, and until we can basically get up to 90 into the 90% tile of the population, so that then we can develop true herd immunity. So that's the deal there. I read in the paper this morning, actually, that Operation Warp Speed is going to be getting ready to set up the largest population areas to get the vaccine first. And that is a bit of a departure. Usually we really have to get it out to the most at-risk populations first, but we'll see. We'll see how that works. I read a little bit, well, I read a couple of misconceptions about herd immunity in your assignment for when you listened to the This Week in Virology podcast. And I just want to reiterate, herd immunity is the minimum percentage of the population that has to be somehow immune to a disease in order to protect everybody. The minimum percentage of a population that must be immune to a disease either through vaccination or immunity that persists from having had the infection, this is getting to be a long definition here. The minimum percentage of a population that must be immune to a disease, either through vaccination or immunity that persists from having had the infection, in order to lower the incidence of the disease, or rather in order to lower the incidence of a disease, to eradicate it and to protect those who can't be vaccinated. So to eradicate the disease and and or protect those who can't be vaccinated. And those who can't be vaccinated, people who are immunocompromised, who have autoimmune disorders, who pregnant women, for example. So it's not only vaccination, but but you require some sort of immunity that either usually comes from vaccination, but immunity can persist from having had the infection. The problem with developing herd immunity for COVID is that immunity does not seem to persist very long. There's a lot of evidence showing that antibodies only last about 4 months. I mean, we're talking immunity that has to last many years. So long-term immunity that persists from having had the infection, and that's kind of wishy-washy. Doesn't happen that often. So this part here that I've underlined here, it's uncommon. It's uncommon, so you really need it through vaccination. Yeah, so that's why that whole Great Barrington thing was just never going to work. Like, hey, let's just let everybody get sick from it, and then we'll all be immune to it, and we can all go back to our lives. But the thing is that immunity doesn't even last that long. We don't know how long that immunity even lasts, and that doesn't even take into the account the horrible outcome that millions of people would die from this. I mean, we're already well over 200,000 deaths from this in this country, and I certainly don't want that to be. I don't want to be one of them, and I don't want any of you to be one of them, or anybody I care about to be one of them. So that's why that whole Great Barrington thing is just not going to work. We have take-home message: we have to get a vaccine, and we have to get it. We have to distribute it in the most efficient way possible. All right. So let's see. We have talked about inactive whole agent vaccines, protein subunit vaccines, toxoid, polysaccharide vaccines, conjugate vaccines, messenger RNA vaccines. All of these here are inactivated vaccines, and they all have low risk. They cannot cause an infection because it's not a whole live virus. Only a live virus can cause an actual infection. So all these babies here, they are low risk vaccines. We already talked about how don't usually require refrigeration. Don't usually, but these 2 messenger RNA ones definitely will. So that's not really much of a pro anymore for inactivated vaccines. Protection does not tend to last as long. You often need booster shots, and as far as booster shots for the new COVID vaccines, we'll see. People have to be monitored to see how long their antibodies persist. So that will be an ongoing sort of thing. All right, we talked about this already. Let's talk about vaccine safety. Okay, we've already talked about herd immunity. Only works if a high percentage of the population is vaccinated. For something like measles, which is wildly contagious, you need something like 97% of the population to be vaccinated against measles in order to reduce incidence of those. I'm well aware of anti-vax rhetoric that has been around here. I have friends who still think that vaccines cause autism. They don't. They don't, and that has been shown time and time again. The person who published this, Andrew Wakefield, falsified his data. He lost his medical license in Great Britain as a result. He was paid to do this, falsified his data for 11 patients that were in this quote-unquote study of his. However, that is not to say that vaccines are without risk, because all medical procedures do carry a risk, and one of the biggest ones are adverse reactions to other ingredients in the vaccines. Okay, so for example, egg allergies. People with an egg allergy cannot have a vaccine that has been grown in fertilized chicken eggs. A lot of live vaccines are actually grown that way, because a virus, to make more of a virus, it has to infect a cell. They actually use chicken eggs for that. If you've got an egg allergy, can't have that. Let's see. There is a class of molecules that I should talk about called adjuvants. Why did I do that? I didn't need to take another screenshot there. Adjuvants. Adjuvants are molecules that are added to vaccines to enhance the immune response. When modern vaccines were first developed in the 50s and early 60s, different companies were producing them, and there were certain companies that were like, okay, we're going to scrub out all of the vats and everything and all the containers that we keep everything in. We're going to keep everything really super, super clean. And the things that were kept super, super clean in a certain way actually ended up producing a less effective vaccine, that there were actually some substances that needed to be in them that, I don't want to use the term dirty, that made them a little dirty, but it removed substances that were getting into the vaccines that actually enhanced the vaccine's immune response. A lot of these are aluminum salts. And also, I mean, now everything is very, very controlled, and these are deliberately added squalene compounds are often added. And what they do is they trigger a more intense inflammatory response in the area of the injection. So you get a stronger inflammatory response at injection, and that stimulates a stronger full immune response. There we go. So adjuvants are deliberately added to vaccines. They are substances that are not intended to have antibodies made against them, but they intensify the inflammatory response and basically call the rest of the immune system on over to that injection site. They are very carefully added now. It is not a haphazard thing as when they were first discovered, basically. And they have been well studied. A lot of anti-vaxxers who still think that vaccines cause autism are in response to a substance called thimerosal. Thimerisol. It's a mercury containing. Oh, sorry, that's magnesium. Hg is mercury containing. Substance that actually used to be a part of some live attenuated vaccine preparations. Emphasis on used to. And this is it's just trace amounts of mercury, but it used to be a part of some live vaccines, but thimerosal has been out of use for quite some time now, like 30 years, so it's not in. I think there's only one vaccine that I think it might be in, but they even took that out a while back too, so it's very, very uncommon, not used anymore, except for maybe one type of the live flu vaccine. But so people who are saying vaccines cause autism because they have thimerosal in them, you can just look that up pretty easily and say, nope, this one does not have it. So I'm going to leave it there, and this will be enough for the week between this lecture. I'm putting, I I put just one really large lecture together, and no, I did not expect you to listen to the whole thing all at once, but this will be enough just for the week. I'm trying to just do one upload this week and make it a little longer than usual. I will see you next time, and I hope you have a safe week, and we'll talk in Zoom. Be well.